everyone, welcome back to Equity Tutors, the budget-friendly biology tutoring service. Today we're going to continue on from Lauren's lesson last week, which was on genetic diversity. She explained in that lesson how mutations occur, how these lead to adaptations and natural selection, how those adaptations and mutations can give you an advantage. Today we are going to look at species, taxonomy and biodiversity. So specifically, we look at how different species are named, the different categories of species, the biological classification. So that includes the binomial system of naming species, the features used to classify organisms into different kingdoms, and then we'll go on to talk about biodiversity, so biodiversity within communities, how biodiversity can be considered different levels, and also how we measure biodiversity, so things like species richness, species evenness, and how these can be assessed, including some examples of calculations. So let's start then with species and taxonomy. So two organisms belong to the same species if they are able to produce fertile offspring. So that is the definition. So courtship behaviour is a necessary precursor to successful mating. And the role of courtship in species recognition is important because it ensures that the individuals of the same species mate and produce viable offspring. For example, courtship behaviour can involve specific displays, vocalizations. Think of birds, for example. So they want to attract another bird of the same species so that their offspring are viable. So what I mean by that is that those offspring can then themselves create more offspring, so they're fertile offspring. So the type of courtship behaviour can include displays, vocalizations, pheromones that are unique to that specific species. So it's kind of like a tell to the other member of that species that they are the same. So classification is the process of naming and organising these species organisms into groups based on their characteristics. So the most common way that this is done is a phylogenetic classification system. So this attempts to arrange species into groups based on their evolutionary origins and relationships. So it uses a hierarchical system, so you have 7 different steps in which smaller groups are placed into larger groups. So what I mean by that is started at the top, so looking at the image you have on screen if you're watching a video, or if not, if you're listening to a podcast, we link the images below. Starting at a kingdom, that is the top level. When we go to level 2, which is the phylum, that is a smaller group of animals. We go to class, that is again smaller group of an organism. Order is a smaller group again, etc., etc., reaching the bottom level of that tree, which is species, which is the most specific and the smallest group. So it's a hierarchical system in which smaller groups are placed within a larger group, with no overlaps between the groups. Each group is called a taxon, or plural is taxa. And as I said, the order of this taxonomic hierarchy is kingdom, phylum, class, order, family, genus, and species. So this sounds like a lot of words that you need to memorize. Unfortunately, that is the case. You do not need to remember any specific examples. They usually will give you that information in exams. But if you want some hints and tips on how to remember this order, I really recommend that you listen to all of our follow-up lessons. I always give you good exam techniques and good ways of remembering things like this that are just essentially a memory test. But I just want to break it down into an example for you. So using humans as an example. So we are a species, and our species, the official scientific name is Homo sapiens. So this is a binomial naming system. So each species is universally identified by a binomial consisting of the name of its genus and the species, such as Homo sapiens. So sapien is the species, Homo is the name of the genus. So this has many advantages. It's really simple, it's universal, and it's consistent. So it provides a standardized naming system that allows scientists across the world to communicate clearly and avoid confusion, as each species has a unique name. So what this means is, whereas in English, for example, we call our species human, this may not be true in different parts of the world, but by using this universal language, this binomial system, scientists everywhere will all refer to humans as Homo sapiens. So let's climb up the ladder using humans or Homo sapiens as an example. So as I said, the next step up on the phylogenetic hierarchy is a genus, and we know from what I've just mentioned that for humans or Homo sapiens, the genus name is therefore the first name in our binomial system, which is Homo. The family that we belong to is the hominids. So these are all primates with relatively flat faces and three-dimensional visions. The next level up is the order. So this is we belong to primates. So you can start to understand here how this is relating to the evolutionary origin. So our closest cousins, our closest ancestors that we belong to in evolutionary terms are monkeys. So they also belong to the same order as we do, which is primates. The next level up again is mammals. So again, we're going to have more things in common with other mammals than we will to, for example, fish, which would be in a different class. And then the next level up is chordates. This just means animals with a backbone, also known as vertebrates. And then finally, our kingdom is animals. And so from here, what I want to do is start to talk about all the different kingdoms that can define all of the species and organisms in the world, because there are a couple of different ways or strategies towards classifying all the organisms, and they all have their advantages, disadvantages, and a lot of similarities as well. So there's a lot of overlap. So remember, we're starting right at the top of this hierarchy. So this is where there's the broadest level of categorization. So imagine before we knew about DNA, before we had sequencing, if you were a scientist a few hundred years ago trying to categorize all life on Earth, how would you categorize the different types of organisms? So this is where the first example of categorization comes in, or classification of the species, and this is using 5 kingdoms. So kingdom is the first level on the tree. So the 5 kingdoms are Monera, Protista, Fungi, Plantae, and Animalia. So this is mostly based on the features of those animals, so the things that you can visually see in those organisms or plants. So for example, Monera. This kingdom is unicellular prokaryotes that lack nucleus and other membrane-bound organelles, and they're basically classified on their cell wall composition and other morphological features. We then have Protista. So this is unicellular eukaryotes that have a nucleus and other membrane-bound organelles. These are classified based on their mode of nutrition, locomotion, and again the cell wall composition. So the first 2 kingdoms that we have are both unicellular organisms, and you already know the difference between a prokaryote and a eukaryote. The next kingdom is fungi. So this kingdom includes multicellular eukaryotes that have a nucleus and other membrane-bound organelles, and they are classified based on their mode of nutrition, cell wall composition, and other reproductive structures. Next, we have plantae or plants. Again, these are multicellular eukaryotes. They have a nucleus, they have other membrane-bound organelles, and technically what they are classed on is their mode of nutrition, cell wall composition, and reproductive structures, which vary from fungi. And then finally, we have animals. And again, these are classified based on their mode of nutrition, body plan, and developmental features. These would be kind of an obvious way to classify all the organisms on Earth without knowing anything scientifically about the DNA. However, as science has advanced over the years, what we now understand is that possibly some of the species within these kingdoms are probably more connected than we thought. In evolutionary terms, there's a lot of overlap between those kingdoms, which doesn't necessarily fit with this hierarchical system that I described earlier, where all of the groups at each level were distinct from one another. So, bear with this in mind, there has since been proposed a new system which recognises 3 primary domains and branches of life. These are archaea, bacteria, and eukarya. So, archaea includes organisms that were once considered to be part of the bacteria kingdom. So these are unicellular organisms that live in extreme environments, these such as hot springs, deep sea, or salt flats. They have a unique cell wall structure that is different from both bacteria and any other eukaryote, and their genetic material and biochemistry is distinct from other organisms. So essentially, we have found a new species of single-celled organisms that look completely different to the other classifications that we had before within the 5 kingdoms. And scientists now believe that these single-cell organisms deserve a kingdom of their own. So the bacteria domain in this new 3-kingdom system will contain all of the other prokaryotic organisms not included under archaea. So these organisms are ubiquitous in the environment, can be found in soil, water, inside other organisms. This will include most of the bacteria that we are familiar with in our everyday lives. So they have a much simpler structure to the archaea and do not have membrane-bound organelles. They are the most numerous and diverse group of organisms on Earth. And then finally, we have eukarya. So this includes all organisms that have a true nucleus and membrane-bound organelles, such as the mitochondria and chloroplasts, and it actually includes plants, animals, fungi, everything that we discussed before, they've put into one group. And again, this is actually because although these organisms can look very different and act very different just from the visual characteristics, on a cellular level, we probably have more in common with the plants, and fungi have much more in common than, for example, the archaea and the bacteria do, which seems crazy when you're just looking at them, but on a genetic level, they are more similar. So just as a comparison and a contrast of this 3 kingdom system and the 5 kingdom system. So the 5 kingdom system is an older system of classification which divides all living organisms into 5 kingdoms based on observable characteristics such as cell culture, cell structure, and nutrition. So these 5 kingdoms are Monera, Protista, Fungi, Plantae, and Animalia. On the other hand, the 3-domain system is a more recent classification that takes into consideration genetic and molecular similarities. So these 3 domains are bacteria, archaea, and eukarya. So the main similarity between the 2 systems is that they both aim to classify all the living organisms. However, the 5 kingdom system focuses more on the observable characteristics, while the 3-domain system focuses on the genetic and molecular similarities. Another significant difference is that the 3-domain system includes archaea as a separate domain, which was not even considered or known about when the 5-kingdom system was established. So archaea was previously classified under the kingdom Monera, but really they were not studied as much at that time, so we didn't really understand that they were their own separate thing. And additionally, the 3-domain system also separates bacteria into a separate domain, while the 5-kingdom system categorizes them again under the kingdom Monera. So overall, the 3-domain system is considered more accurate and comprehensive than the 5-kingdom system because it accounts for the genetic and molecular similarities, which provide more precise information on the evolutionary relationships between organisms. But personally, I think it's a little harder to understand on a more surface level. You think, how can animals and fungi be more evolutionary related than the bacteria and the archaea? It's a little harder to get your head around, but believe it or not, genetically they are closer together. So now I want to move on to talk about biodiversity. So biodiversity is a term that encompasses the variety of life on Earth. So we're talking about all the different organisms and species that exist, and what we are going to get into in this lesson specifically are terms to define biodiversity, how biodiversity can be considered at different levels, and even how we measure diversity, how it can be assessed, including some calculations. And you may be familiar with the Simpson's index of diversity. To calculate a biodiversity, this is a very common exam question. You do not need to know the formula to calculate the Simpson's index of diversity, but you do need to know, based on information or data they give you, how to calculate using the formula they provide and how to interpret that result. So, biodiversity, as I said, is a term that encompasses the variety of life on Earth. It includes a variety of habitats, ecosystems, and species that exist, as well as genetic diversity within species. So firstly, the levels of biodiversity. Biodiversity can be considered at different levels, as I said, habitat, species, and genetic diversity. So what do I mean by habitat biodiversity? So this refers to the diversity of different habitats on Earth. So how do the different places that organisms live vary? So for example, sand dunes are very different to woodlands, different to meadows, different to streams, and there are different species living in all of those different habitats. So a habitat is referring to the place where an organism lives. Species biodiversity can be measured using species richness. So that refers to the number of different species in a community, and species evenness, which is the number of individuals within each species. So when we talk about, for example, the rainforest being one of the most diverse places in the world, what that actually means is there are a lot of different species there. So that means species richness. However, if you were to look at, for example, a big city like New York, that wouldn't have species richness. What it would have is species evenness, which is the number of individuals within that species. And within the species, you will have biodiversity within that group itself. And then genetic biodiversity refers to the diversity of different breeds within a species and can be measured by calculating the proportion of polymorphic gene loci. So we know from previous modules that a loci is just a location in the DNA. So when we refer into genetic diversity, and I'm saying that we can measure the diversity of different breeds within a species using gene loci, what I'm saying is how different is that location in the DNA within the species. So if I look at chromosome 5 at location X in 100 people within a certain area, how much is that location going to vary from one person to the other? So that's what I mean by the genetic variation in a gene locus. So this is just one way of calculating the genetic diversity. So the way that you calculate that is a percentage. So the number of people who have a polymorphism in that location or polymorphic gene loci over the total number that you've measured. So for example, if a population has 100 loci and 20 of them have the variation in the allele, then the proportion of the polymorphic gene loci would be 20 over 100 or 20%. So we can use that calculation to compare genetic diversity of different populations or to even monitor changes in genetic diversity over time. And suitable populations for this type of analysis is you would look at animals in zoos, captive breeding programs. For example, in exams, you should be able to interpret data relating to the similarities and differences in the base sequences of DNA and in amino acids relating back to different organisms and genetic diversity. You do not need to recall any of this information. They will give you sample data sets. So for example, they will say there is a breeding program within a zoo and we want to look at what is the genetic diversity within these animals at the zoo, because it is important to maintain genetic diversity. So they may look at the proportion of polymorphic gene loci over time. So they may give you data from 1990 and then 2020 and ask you to compare and contrast the genetic variation then and now. So you would need to be able to calculate the proportion of animals within the data set from 1990 and from 2020 that have a polymorphic locus, so a change in their DNA at that location, and then reflect on how that has changed over time and what this means for the genetic diversity of those animals in captivity. We go over practice questions like this in the bonus lesson over on Patreon page. Now, to go back to the different kinds of genetic diversity and how they are measured, so not just genetic diversity. If we want to measure, example, species richness in a field, we have different sampling techniques that we can use in order to measure the diversity in that field. So we have random and non-random sampling techniques. Random sampling involves selection of a random location within the habitat, so a random place in the field, and you collect data from that location. So you select a random square foot in the field and look at how many, for example, worms there are in that location. So this technique ensures that every part of the habitat has an equal chance of being sampled and reduces bias. Then you have non-random sampling techniques. So these are selected based on the specific characteristics of the habitat of the organism being studied. So these can include opportunistic sampling, stratified sampling, and systemic sampling. So opportunistic sampling, for example, is collecting data from locations that are easy to access, such as areas near the edge of the habitat or where there's higher density of organisms. So you can see how these non-random sampling techniques would bias the data a little bit more. Stratified sampling involves dividing the habitat into different areas or strata based on specific characteristics such as soil type or vegetation cover, and then sampling each stratum separately. So for example, you take a very wet part of the field, a very dry part of the field, and see how they would vary. And then systemic sampling involves selecting sampling locations at regular intervals, such as every 10 metres along a transect line. So this kind of combines different methods, whereas it's a non-random approach, but you are making sure you cover a diverse range of that habitat. And so each of the techniques has its advantages and disadvantages, and the choice of which technique depends on the specific research question. So just be prepared. So for, again, the type of questions where they present you data, they explain to you an experiment that has been done, be prepared to identify, based on the strategy used, what are the disadvantages and advantages based on the specific research question. But remember that it's always essential to sample a range of organisms in a habitat to get an accurate picture of the whole biodiversity. So next I want to talk about the index of diversity. So this is a measure of the relationship between the number of species in a community and the number of individual in each species. So it's the richness and the evenness. So to calculate the index of diversity, we use something called the Simpson's index of diversity formula. So that has a complicated formula which is on screen now. As I said, you do not need to memorize. What you do need to know is N is the total number of organisms in all species, and the little n is the total number of organisms for each species. So the higher the index of diversity, the more diverse the habitat is. So it's taken into account the species and the richness in order to get an overall picture of the diversity. I recommend that you practice these questions. So they'll give you values for the total number of organisms and the number of organisms in each species. You just need to plug those numbers into the formula and it'll give you a diversity index. And as I said, the higher the index, the more diverse the habitat is. So finally, to end the lesson, I want to talk about why biodiversity is important and some of the conservation and farming techniques and efforts that are being made to maintain biodiversity. So firstly, why is it important? So it's critical to the function of the ecosystem, to the world. So ecosystems rely on the diversity of species and the interactions between them to perform important ecological processes. So every animal, every species, every organism is doing a job. It's all in the circle of life. So such as nutrient recycling, pollination and decomposition. And without the biodiversity required for all of this, ecosystems would become less resilient and we wouldn't be able to adapt to changing environmental conditions. Secondly, it's important for human health and well-being. So many of medicines that we use today are from natural resources, and biodiversity is essential for the continued discovery of new drugs and treatments. Additionally, ecosystems provide important services such as clean air and water, which are essential for humans to survive. And finally, it has economic value. So many industries such as agriculture, forestry, fisheries depend on biodiversity for their livelihoods, and even ecotourism is now an important source of income to many countries, and that relies on preservation of our biodiversity. So overall, it's important for the function of the ecosystem. Essentially, if we lose biodiversity, it's really, really bad for the environment. Things would impact humans very quickly if we lost biodiversity. So that is the reason why we have all of these ways of measuring genetic diversity. And so I just want to touch on one example of human impact on genetic diversity or diversity in general. And that is farming. So farming techniques will reduce biodiversity. So of course, farmers want to produce the best produce to sell to consumers, and often that means having the animals which will make the most milk or the most of the product that they are trying to sell, and that often comes with the animals being very genetically close to one another, so genetic diversity is lowered. And that is measured by looking at the frequency of measurable or observable characteristics, the DNA. If you look at for example, the difference in the polymorphisms of loci, that will be very low in farmed animals. And this is because they have been bred that way, number one. And secondly, because they've removed from their natural habitats, they no longer need to adapt and survive. They are being farmed. So farming is one example of how humans are having a really bad impact on genetic diversity. On the flip side, there are some great conservation efforts. So these can be in situ or ex situ. So what I mean by that is in situ in the organism's habitat, ex situ outside of the organism's habitat. For example, ex situ captive breeding programs. So imagine in a zoo for example, a panda, which are endangered species, and they set up breeding programs for those pandas ex situ, so within the zoo, in order to for the pandas to breed and hopefully maintain some genetic diversity. So endangered species are carefully bred to increase genetic diversity and population size. Genetic diversity is maintained via the exchange of organism and gametes, keeping stud books and preventing inbreeding, and use of techniques such as IVF. There are reintroduction programs which aim to release animals bred in captivity back into their natural habitats. So the animals being back in their habitats will naturally lead to adaptations, and breeding in those natural habitats will increase diversity. And then seed banks. So this is a storage of a large number of seeds to conserve genetic diversity and prevent plant species from going extinct. So storing seeds instead of plants means that there are a large variety of species that can be conserved. This is cheaper than storing the whole plants, obviously, and takes up less space. So the plants are just stored in cool, dry conditions to maximize the amount of time they can be stored, and they are periodically tested for viability. There are then some in situ conservation methods, things like education programs. So educating people in those communities about the importance of maintaining biodiversity, and breeding programs and illegal trade of animal products. You may get to experience visiting countries that are really good at this kind of things, countries like Costa Rica, where they really look after their animals, and they have people who are on call to come and rescue animals that may have escaped from their natural habitat into communities, and the people in those communities really respect the animals because of good education programs, know who to call, and the animals will be taken back to the natural habitat. And initiatives such as national parks and sites of specific scientific interest, which aim to conserve these habitats and biodiversity and leave them untouched by humans. So just to recap today's lesson, we covered species and taxonomy, how different species are named according to their evolutionary history, and a comparison of the 5 kingdoms versus 3 kingdoms way of classifying organisms. We then spoke about genetic diversity, ways of measuring genetic diversity, and why genetic diversity is important and what efforts are being made to maintain genetic diversity. So thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!